Hello guys, welcome to the first episode of this channel. I'm Adit and today we will be considering class 12th genetics unit. Now I will start the syllabus from this and moving on afterwards. So in this unit we will pick today's chapter as principle of inheritance and variation. And in this we will be you know getting deeper into Mendelism today. So let's get started. Before we start Mendelism, there are certain things that you need to understand and clear those definitions first. So let's take a look. So the term genetics, what does this term means? This means the study of heredity and variation. So, and what does these term heredity and variation means? Well, heredity terminology denotes to the passing on of genetic character from parents to the offsprings, and variation is the differences in each and every individual of same species. So these two things are related together, and when we study them together it is called genetics. So it was easy and simple. Now some scientists regarding the genetics are George John Mendel. As you know, we will be studying Mendelism today, and he is the father of genetics. W. Bateson. Now he is the father of modern genetics. Thomas Hunt Morgan. Now he is the father of experimental genetics, and you may all know his famous experiment on Drosophila, and with that experiment he tried to explain linkage and crossovers and many other things like that. A. Garrod. Now he is the father of human genetics and biochemical genetics. Now why he is known for human genetics? Because he was the first to discover a human metabolic genetic disorder known as L-captonuria, which is commonly called black urine disease. Now, in this disease, the enzyme homogenetic acid oxidase is in less quantities, which causes this disease, and he named the whole concept as one mutant gene, one metabolic block. Now we will be moving to some deeper terminologies, and the first one is factor. Now before we talk about these definitions, let's go back and see what does we really mean when we talk about the term genetics. Now in this we generally regard to genes and their interaction. Now what is genes? Genes are the pair of codons, not a pair, group of codons, in a sequence which express one or more character in our phenotype or genotype as well. So the genes express a character. Now the character can vary from individual to individual. This depends on allele. Now allele are different types, or you can say the variation in a gene. Consider an example like eye color. Now it can be black, it can be brown, it can be green, it can be blue. So we are talking about a same gene here, but as you know, the colors are different. This means there must be something that vary in these genes. So yes, it is the alleles. Think like alleles are the different version of same gene. Now in a gene we talk about a big thing like the eye color, but when we go deeper there are allele of that same gene. Like there will be an allele for the black color, a separate allele for the blue, same goes on for brown and green. Now the individual having the black allele of that gene will have black eye colors. Now this concept was not known when Mendel performed his experiment, and he named those as factors. And what does he mean by factor? Now factors were the basic unit of heredity which gave a character to an organism. Basically he was talking about gene, not knowing what he was talking about, because back at that time gene and allele concept was not discovered. Now later on these factors were named genes by Johansen, a scientist in 1909. But Mendel used the terms like factors or element for these. There were made improvement in these factors and they were first represented in letters by the scientist named Morgan, and he used capital letters to define the dominant trait, and so he used small letter to define the recessive traits. Now we were talking about alleles. Now what are alleles? Alleles are the different form of gene, or you can say variety of genes which are present at same point on a homologous pair of chromosomes. The term allele was coined by Bateson. Now we have come across another word, which were the dominant trait and the recessive trait. Now what does these means? Basically, the zygote is formed by the fusion of two gametes. Now for a specific trait, let's consider the eye color again. If both the gametes were having the same allele of a gene, then it will be called the homozygous pair. Okay, so homozygous means, homo means same. So if both the traits are dominant or recessive, then it will be called the homozygous. And the opposite case is called the heterozygous, when a zygote is formed by the fusion of two gametes having two different allele of same gene. Yes, one is dominant and one is recessive. Now this case is known as heterozygous. Now there is another term which is known as hemizygous. When a zygote is formed by the fusion of two gametes but have only one gene in a pair, means instead of having two alleles, it has only one. For example, you can take male humans as hemizygous in terms of sex-linked genes, because they does have only a single Y chromosome which just have one allele of a gene. There is no second to complete the pair. So I hope you find this very easy. Now if you do not understand these, I will be moving on, and further you will get to see how these are implemented, and then you can see how easy it is. Now I have used two terms you may have noticed. They are phenotype and genotype. Now what are these? Phenotype is the character which we can observe, the morphology of an organism, and the genotype is the genetic makeup of that organism, which we cannot see from our eyes but can be known by a set of experiments. So these are two things, genotype and phenotype, and these two will be used intensively in Mendelism. Now again, genotype and phenotype, both these terms were coined by Johansen. Remember, the guy who named factors to genes. Now there is another term related to phenotype and genotype. It's phenocopy. Now what does this mean? If two different genotype organisms are placed into different conditions and they produce same results, now their phenotype is same, then these two organisms are known as phenocopy of each other. So now we are all familiar with the terminologies which we are going to use in the topic Mendelism. Let's start with it now. Now what does the term Mendelism means? Basically, Mendelism stands for the experiment performed by Mendel in the genetics field plus the description of the conclusions of those experiments plus the principle on which Mendel explains the heredity. These all three things when come together defines Mendelism. So now let's just start. So Mendel performed several experiments on the garden pea, scientific name Pisum sativa, and he has concluded many of laws based on probability and his experimental work. Now George John Mendel was not recognized for his work until these three scientists came into existence. Their name Carl Coren. Now he experimented on the plant maize. Hugo de Vries, he experimented on evening primrose. Eric von Schermack, now I'm not sure if it's pronounced like this, but he experimented on different plants. Now these three scientists rediscovered Mendelism after 34 years when Mendel has died. So basically, they rediscovered Mendelism, and the three laws which we study are based on the rediscoveries. Now, the first law we study is law of hereditary inheritance, Mendelism. These three are the same thing. They were given by Mendel. The next two laws were not given by Mendel. They were given by Carl Coren, and they are: first, law of segregation; second, law of independent assortment. Now let's talk about the first law, the law of dominance. Now what this means: one out of the two factors or allele representing a factor is able to express itself as dominant character. Now what this all means is that in the pea plant, let's say we consider a factor tall. Now according to Morgan, capital T defines a tall plant while a small t defines a small plant allele. Now if in a heterozygous condition where there is a zygote formed by fusion of two gametes, one of the gametes was having the capital T trait while the other was having a recessive small t trait. Now the gamete formed is a capital T and a small t, so the capital T will be able to express its characters as a tall pea plant, while the small t will do nothing and sit there idly, will not be able to dominate. And from here the terminologies comes dominate and recessive. So this law was pretty cool to understand, and it's just very easy. Now if you have a zygote having both the T capital, now the both T will not express the character and the plant will be gigantic. No, only one of those will express its character and the plant will be just normally tall. But for a plant to be short, you should have a homozygous recessive condition, means the trait which is not able to dominate should only be present in both the cases, like this one, homozygous recessive plant where both the pairs are small t and small t, then the plant will be a short plant. Let's take a very short example here. Consider two plants with one having homozygous tall and the other is homozygous short, which means their pairs will be like capital T and capital T and small t and small t. When you crossbreed them, the F1 generation produced are 100% heterozygous, means all the plants are phenotypically tall but they are heterozygous when you consider the genotype, so they will be capital T and small t. Yes, so here you can see the law of dominant. The capital T, the dominant trait, is able to express itself while the small one does not express itself at all. Now let's go to the second law, which is law of segregation. Now what does this mean? That the two contrasting alleles of a same gene does not get mixed up, rather than they separate during the formation of gamete. So this was the whole concept. So the gamete carries only one allele of a gene and the alleles do not get mixed up. Now to experimentally prove this, Mendel used some probability. Now don't get scared, it's not tough maths. The first example which we took was this one. Now if we try to draw a Punnett square around here and do the same test as accordingly, we see that the F1 generation is all heterozygous tall. But hey, why wait here? Cross two of those and we get this chart. Now here we can see when two heterozygous tall plants crossbreed with each other, we get a ratio like 1:3:1, where 1 is homozygous tall, the 2 are heterozygous tall, and the 1 at last is homozygous recessive. Now what's this? This is a phenotype ratio, and every time Mendel performed experiment on thousands of the pea plants, the results were approximately the same. And why I said approximately? Because this is a mathematical situation where everything is ideal, but in the case of Mendel, he was working on a real pea plant. All the seeds were not captured and all of them do not grow up into a plant. So many of those results were taken, the observation were taken, and the average was always the same. And the result was same because the traits, these two traits, they got separated out and do not mix with each other. So this proved the law of segregation. This law has some limitations now, which we know, and those limitations are four basically. The first is incomplete dominance, where the dominant trait is not able to express the character at its full strength. There is some interference from the recessive trait, like in the flower color of Mirabilis jalapa. Now, the basic colors, the pure colors are red and white, but when we try to produce an F1 generation, the colors should be red according to Mendel and its laws, but the color is pink. Here we can see incomplete dominance in word, where the dominant character is expressed, but there is some interference of the recessive character too, which does not allow the dominant character to attain its full strength. The next shortcoming was codominance. Now in the codominance, there are many alleles present of a same gene or a factor, but we know that only one, the dominant trait, can express itself. But in this case, both of them express themselves at full strength. For example, take a look at the blood group. It's A type, B type, AB type, and O type. Especially in the AB type, we have alleles for both type A blood group and type B blood group, and they express themselves at full strength. So here both are dominant, both are different, and both express themselves at full strength. So this was the example of codominance, multiple allele, and the example which I gave in the codominance is pretty much the case with the multiple allele. It goes the same way. There are many alleles of a given gene and more than one of the allele are dominant. And the last shortcoming was the pleiotropic genes. Now these genes are basically one allele or one character of a gene controlling many traits, like the gene A controlling many characters which appears in phenotype. So wherever this gene goes, it seems like many genes are going together at the same time, but it's one gene expressing many characters out there. So these were some limitations to the law of segregation of Mendelism. So you got that, right? Let's move on to the third and the last law, the law of independent assortment. Now this law is rather big one. What does this law say? This law says that the factors of the genes are independently separated from each other and are randomly arranged in a gamete, there is no repetition of them. The offspring gets one of the allele which is random. So let's take a look at the Punnett square again, but this time we are not having the monohybrid cross, we are going for a dihybrid cross, so you need to pay attention. Now before we take only single character which is tall, now we will take the seed shape and the seed color of the pea plant. Now the seed shape is round or wrinkled, we will represent round with R and wrinkled with small r. And the color is either green or yellow, so we will represent green with capital Y and yellow with small y. Let's arrange. So first of all, let's start from beginning. We take two parents, homozygous dominant and homozygous recessive. We cross those parents, get all the possible combinations of the gametes or the offsprings. Now we cross these offspring in the dihybrid cross. Now we are crossing the F1 generation to get F2 generation, so have a good look at that. We arranged everything accordingly and started multiplying. It's very simple, as you can see here, and when this is all done, we take out a phenotypic ratio of each and everything arranged here, which is 9:3:3:1. Now the 9 is the ratio of the rounded and yellow seeds. The 3 denotes to rounded and green seed. The next 3 denotes to wrinkled and yellow seeds. The last denotes to wrinkled and green seeds. This was the phenotypic ratio every time Mendel performed this experiment, and he came to a conclusion that the shape of the seed and the color of the seed are independently assorted to the gamete at the time of gametogenesis. Now he was right at that time, but not far away Morgan performed the experiment on Drosophila and proved linkage. Now Mendel was rather lucky guy who picked up two characters which were not linked with each other. What would have happened if he had put Drosophila in case of P and he got linkage in the characters? Then this whole experiment was proved to be wrong, and this was the criticism factor, linkage. Morgan find linkage, and he gave this idea that two characters which are placed very close to each other on the DNA are linked to each other and have a great chances of not getting separated from each other at the time of gamete formation. It's not like the Mendel has just performed the experiment with the shape of the seed and the color of the seed. Mendel has experimented with all the 7 characters of the pea plant. He was so lucky that none of those 7 characters were actually linked with each other. So every time he performed the experiment, the results were 100% accurate. Now let's talk about the success of Mendel. How he was so lucky. Now the first thing was Mendel only took 1 or 2 characters of the pea plant rather than picking all of them at the same time, which his predecessors have done. The same mistakes have been done, taking many characters at the same time, and they got into codominance and multiple allele systems and linkage and blah blah blah, everything of hurdles and they were not able to do anything good. Now the second reason is selection of material. The pea plant, now the lifespan is short, has only less character, so you can consider 1 or 2 of them easily. The characters are very distinguishable from each other. The pea plant is also naturally self-pollinated. Now the flower doesn't open and the pea plant is self-pollinated, so he doesn't have to take in consideration of the cross-pollination and everything, and he can decide whether to perform a cross-pollination or not and definitely get good results during cross-pollination, as we can see in the hybrid. Now, the pea plant seeds are large, and Mendel just did not work on the pea plant. He also worked on honeybees and rajma. Remember that. Now, the third point is Mendel quantitatively analyzed the result and the outcome of his experiments, and he used probability to map down all these things, including some maths in biology. And the good thing was he was not lazy. He maintained all those paperwork in a very good condition, and he kept on making records of everything he saw and he observed from his experiments. Here is the list of things which you might consider helpful or not. I don't know, but here's a list. You can take a look at it, all these 7 traits, and what they their chromosome number on which they are present, and the height, and all the measurements and stuff like that. You can get it here. Take a look, pause the video and take a look there. Now, there were some bad points about P also. Remember pleiotropic genes? Yes, one gene which define more than one character. The flower color and the seed coat was defined by the same gene, which Mendel cleverly never wrote about. Now let's just have a look at what exactly did Mendel do in order to get those hybrids and everything. So he developed a technique known as emasculation and bagging or hybridization in plant. Now basically what he does, he takes 2 plants which are pure, homozygous tall and homozygous short. Let's say for example, now when the flowers of the tall one were maturing, he just cut all this stigma, or rather say he cut the stigma of one and the anthers of the second, to make sure that they never self-pollinate with each other. To make sure that they never self-pollinate. Now he masked the whole flower with a bag in order to prevent cross-pollination from any uncertain pollen grains. And when the flowers were pretty mature, removed the bag, do the cross-pollination by himself, and got better results, better and accurate results. This whole technique was known as emasculation and bagging. Now separately, emasculation means removing one of the 2 or both the gamete organs. So if you remove anther or stigma from an immature flower, it's called emasculation, and covering the emasculated flower with a bag is known as bagging. It's that simple. So this was it for Mendelism. Now definitely this topic requires some attention, like monohybrid cross and dihybrid cross. There are certain type of cross which are in Mendelism, but let's keep this video this long, or it's very long, I know. Pause the video, rewind it if you doesn't understand anything. If you find anything crazy, or if I have misspelled anything, you can write down in the comment section below. Or if you have any questions, just check the description of this video. I will have my Twitter handle there. You can tweet your question or give the question in the comment section. You can also contact me through the Facebook page where I will upload this video. See you next time, guys.